Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 14 of your OpenSTAX book, Antimicrobial Drugs. I'm Mr. Kennedy, and I'll be your guide as we explore these topics together. Learning outcomes for Chapter 14 include an overview of antimicrobials, the considerations of their use, comparing and contrasting different antibacterial, antifungal, and antiviral drugs, and a discussion involving the factors associated with antibiotic resistance. We begin with a brief history of antimicrobials. Traditionally, antimicrobials are derivatives found in plants. They have been used for decades. Early antimicrobial use began with the idea of chemotherapy as developed by Paul Ehrlich and later Salvarson for syphilis. Chemotherapy is the use of drugs to treat a disease. In 1928, Alexander Fleming identified Penicillium notatum and its active compound, penicillin. The first synthetic drug was sulfonamide. Antimicrobials may exhibit bactericidal or bacteriostatic actions on the microbes they're used against. Bactericidal actions kill the microbes directly. Bacteriostatic simply prevent the microbes from growing. Microbes treated with antimicrobials that create bacteriostasis can still complete their life cycle. The spectrum of antimicrobials. Antimicrobials can be broad spectrum or narrow spectrum. A broad spectrum antimicrobial drug can affect both gram-positive and gram-negative organisms. As an example, tetracycline, which affects gram-positive and gram-negative chlamydia and rickettsia. Broad-spectrum antimicrobials are often associated with the ability to pass through the gram-negative outer layer. Their advantages is that they're more likely to kill an unidentified pathogen. A major disadvantage is that they do not differentiate between beneficial and harmful microorganisms and can increase our chances of superinfection or infection by a second pathogen. Narrow-spectrum antimicrobial drugs affect one type or a narrow group of organisms. This is an illustration showing us how our normal microbiota may keep opportunistic pathogens in check. When broad-spectrum antimicrobials are used, they kill all organisms in a non-selective manner, which can result in drug-resistant pathogens proliferating and causing a superinfection. Dosage of antimicrobials. The goal in dosing antimicrobials is to find a level that produces the desired outcome without adverse reactions. The optimal dose of antimicrobial can be different at different ages and can depend on liver and kidney function. It can also depend on whether the antimicrobial is administered orally or intravenously. The incorrect dosage, or even sometimes the correct dosage, can lead to potential interactions with other drugs. Selective toxicity. Selective toxicity is the idea that drugs can affect some aspect of the pathogen's physiology that is not part of the host physiology, therefore being selective in its potency and its toxicity. Some examples might include an antimicrobial agent's ability to block an enzyme that only the pathogen has in a unique pathway to that pathogen. It may block cell wall synthesis, since animal cells, especially humans, do not have cell walls around their cells. Utilization of antimicrobials that block cell wall synthesis is relatively safe. Selective toxicity may also include blocking translation. The ribosomes in a prokaryote are different than that of a eukaryote, so blocking translation at the level of a prokaryotic ribosome is safe in a eukaryotic organism. This is an illustration of the targets of antibacterial components. Take a moment to pause the video and reflect on each of these before moving on. Cell wall synthesis inhibitors. Cell wall synthesis inhibitors include the beta-lactams, glycopeptides, and bacitracin. Each is responsible for inhibiting or blocking the formation of a cell wall, which may result in a bacterial cell succumbing to osmotic pressure in the host. Beta-lactams all have a beta-lactam ring structure and interfere with peptidoglycan synthesis in the cell wall. Many bacteria produce beta-lactamases, which are enzymes that break the beta-lactam ring, making them resistant. Penicillins, cephalosporins, and carabinums are all examples of beta-lactams. This is an illustration of the functional groups of different penicillins, ampicillins, and the like, and the spectrum of activity. Take a moment to familiarize yourself with each before moving on. Glycopeptides. An example of a glycopeptide is vancomycin. It was discovered in the 1950s and is bactericidal for gram-positive cells only. It is considered our last line of defense for MRSA because it has serious side effects and is expensive. Its use is restricted. Bacitracin inhibits cell wall synthesis in its early stages, is broad spectrum, and works effectively against gram-positive strepto and staphylococci. It's a topical agent, or typically used on skin. This is an illustration of how antibacterial drugs may target prokaryotic ribosomes. Pause the video and take a moment to reflect on this diagram before moving on. Protein synthesis inhibitors. Protein synthesis inhibitors may bind to one or the other of the subunits associated with the ribosome. Aminoglycosides, such as streptomycin, neomycin, and gentamicin, are broad-spectrum bactericidal antimicrobial agents that bind to the 30S subunit, as do tetracyclines. Here we have an example of protein synthesis inhibitors that bind to the 50S subunit. Polyketides, erythromycin, and azithromycin are broad-spectrum antimicrobials that bind to the 50S subunit. Here are a few more protein synthesis inhibitors that bind to the 50S subunit. These are effective against things such as streptococci, staphylococci, and typhoid. They are considered broad spectrum and effective against a wide variety of microbes. Inhibitors of membrane function include polymyxin B and daptomycin. Polymyxin B is a topical bactericidal effective against gram-negatives. It's often combined with bacitracin and neomycin in non-prescription ointments. Daptomycin is produced by streptomyces and is used for skin infections. It only attacks gram-positive cells. Inhibitors of nucleic acid synthesis. Here we have two examples of inhibitors of nucleic acid synthesis. These antimicrobials interfere with DNA replication or potentially block RNA polymerase in bacteria. They can be effective against a wide range of microorganisms and in some cases can also block the growth and subsequent development of protists. Another example of a nucleic acid synthesis inhibitor is listed here. Ciprofloxacin is probably one that most of you have heard of before. Ciprofloxacin is a broad-spectrum nucleic acid synthesis inhibitor, which inhibits DNA gyrase during DNA replication. Inhibitors of metabolic pathways. Antimetabolites are antimicrobials that inhibit or compete for bacterial metabolic enzymes. Sulfonamides are examples of specific antimicrobials that work in this way. They inhibit folic acid synthesis and can be bacteriostatic. They are broad-spectrum antimicrobials. Antimicrobials that target eukaryotes. Antimicrobials that target eukaryotes can create a variety of difficulties in their use. As antimicrobials target eukaryotic characteristics, it is difficult to distinguish pathogen from host, especially in a human. Target cells are similar in this case. There are fewer selective targets, more potential for toxicity, and viruses can be found inside the cells, which makes them difficult to locate and to eradicate. Antifungal drugs. Antifungal drugs are effective against fungi. The cell membrane of fungi contain ergosterol, not cholesterol. So antifungal drugs often make this their target to disrupt the cell membrane and avoid injuring or damaging the host cell membrane. Here's a list of some common antifungal drugs. The inhibition of ergosterol synthesis is done by imidazoles. Binding ergosterol in the cell membrane and creating pores that disrupt the cell membrane is done using polyenes. Antiprotozoan drugs. Antiprotozoan drugs inhibit electron transport in mitochondria. They can inhibit folic acid synthesis, produce damaging reactive oxygen species, inhibit DNA synthesis, and even inhibit heme detoxification. Antihelminthic drugs. A helminth is a worm. Antihelminthic drugs inhibit microtubule formation, reducing glucose uptake. They can block neuronal transmission, inhibit ATP production, induce calcium influx, or inhibit RNA synthesis to kill these pathogenic or parasitic worms. Antiviral drugs. Antiviral drugs affect mechanisms involved in viral infection and replication. Since a virus is not alive, there is no way to effectively kill it, but instead to prevent it from infecting a host and then reproducing once inside. Antiviral drugs target things such as nucleic acid synthesis, the escape from the endosome. They include protease inhibitors, neuramides inhibitors, and agents affecting uncoding. Acyclovir. Acyclovir, or Zovirax, is an analog to guanosine and inhibits replication. It's used for herpes virus infections. Anti-HIV drugs. Anti-HIV drugs fall into a category of highly active antiretroviral therapy. There are drugs that include reverse transcriptase inhibitors, protease inhibitors, and fusion inhibitors that are used in concert to slow and/or stop the growth of HIV. This is an illustration of that process. Cases of drug resistance. Drug resistance is not a new phenomenon. Factors that accelerate resistance include using outdated or weakened antibiotics, using antibiotics for the common cold and other inappropriate conditions, using a subtherapeutic dose, using antibiotics in animal feed, or failing to complete the prescription regime. This is the mechanism for drug resistance. First step is inactivation. Second is the blocked penetration of the drug. Third is target modification. And fourth is the rapid ejection of the drug. Target modifications include ribosome subunits, which provide resistance to macrolides and tetracyclines. Lipopolysaccharide LPS structures provide resistance to polymyxins. RNA polymerase provides resistance to ramphapin. DNA gyrase provides resistance to fluoroquinols. Metabolic enzymes provide resistance to sulfa drugs. Peptidoglycan subunit peptide chains provide resistance to glycopeptides. Target overproduction, or bypass and target mimicry. If the drug inhibits an enzyme, the microbe may overproduce it. The bypass is an alternative mechanism. Target mimicry involves bacteria producing proteins that block the drug. Multidrug resistant microbes and cross-resistance. Organisms that exhibit multidrug resistance or cross-resistance are sometimes referred to as superbugs. They carry more than one resistance mechanism. In cross-resistance, one mechanism can protect against different drugs. Antimicrobial susceptibility test. The antimicrobial susceptibility test is a test in which antibiotics work against certain pathogens. It's useful for therapy and in determining dosage, and mostly done in clinical labs. The Kirby-Bauer disc diffusion test. In a video prior to this, you learned about the disc diffusion test. In this scenario, microorganisms are exposed to a variety of antimicrobial agents that are infused upon discs placed on a petri dish. The effectiveness of the antimicrobial agent is determined by the zone of inhibition that later develops around each disc. Dilution tests. Dilution tests determine the minimum bacterial concentration of an antimicrobial drug. The test organism is placed into wells of a tray containing dilutions of a drug, and then the growth is determined. This is an illustration of a microdilution tray. It would allow us to examine several samples at once. The E-test is a more precise variation of the disc diffusion test. Again, a value of MIC is obtained. The end of antibiotics. It is often questioned or wondered when antibiotics will lose their effectiveness. We are seeing more and more organisms exhibit antibiotic resistance, multi-drug resistance, and there are very few new drugs on the market. So what can be done? We can use the genetic information that's available out there. We can think outside of the box to enhance immunity to attack microbes, and maybe even take advantage of bacteriophage to improve our ability to defend ourselves from potential pathogens. And this concludes our look at Chapter 14 in Antimicrobial Drugs.